So our last speaker of this session is Paula Cushing, and she's going to talk to us about the trophic specialization and chemical integration of newly described spider and symbionts.
All right, thank you very much. So back in 2019, Martin Ramirez and colleagues described a new species of spider, and the colleagues included myself and Norm Horner. This new unusual species was different enough from all the other species that he compared in the study that he and the co-authors decided to place it in its new family, Myrmeciculturidae. And this unusual little spider, Myrmecicultur chihuahuensis, has only been found in association with ant nests. So it's been found in association with ants in the genus Novomessor and ants in the genus Pagonomyrmex. The natural history observations, and there aren't very many prior to the work that I'm going to present today, suggested that the spiders might be myrmecophiles. So the spiders, a fellow by the name of David Lightfoot, observed, he was the only one who observed these live spiders on the nest of a Pagonomyrmex ant colony down in the Chihuahuan Desert in Mexico. And David Lightfoot, he observed this at night. He saw several spiders walking around on the surface of the ant colony. And he seemed to observe what we call pseudo-antenation, the spiders approaching the ants and using their front legs to contact the ants. Previous to David's natural history observations and subsequent to it, the spiders had only been collected from wet pitfall traps placed 1/2 to 1 meter from nest entrances of Pagonomyrmex or primarily Novomessor ants. So Norm Horner and I, my co-author of this study, hypothesized that these spiders might be myrmecophiles, that like other spider myrmecophiles might be living inside the ant nest chambers. And they may be feeding, we hypothesize, on other small soil arthropods that are living inside the nest, like Columbella, proturans, mites. And if this was true, then perhaps they had evolved behavioral and or chemical strategies to allow them to become integrated and associated with these ant colonies. So in order to test these hypotheses, we traveled to the Dalquez Desert Research Station in Texas in the United States. And this is deep in the Chihuahuan Desert. And this is the primary location where these spiders have been documented. So Norm Horner and I and a colleague of Norm's, Jesse Roger, traveled to Dalquest. And what we wanted to do there is we wanted to collect live spiders, which is challenging, but we were hoping to collect some live spiders and use those live spiders to carry out behavioral bioassays using the living spiders and ants from the colony where we collected the spiders, and look at the behavioral interaction to determine if this pseudo-antenation happened, and also to determine, in order to determine if the spiders are picking up cuticular hydrocarbons, we worked with another colleague, Adrian Bruckner of the California Institute of Technology. And Adrian does a lot of work with other myrmecophilic arthropods. So Adrian sent us some deuterated chlorohydrocarbons that we could use to coat the ants from the same nest that we pulled the spiders, if we were lucky enough to get spiders. So we would coat the idea is we would coat the ants with this deuterated cuticular hydrocarbon powder, and we would introduce the ants to the living spiders, look at the behavioral interactions. And if the spiders were closely interacting with the ants, there was a chance that they would pick up the CHC profile of the ants through these behavioral interactions. And then Adrian, the plan was to test whether this was happening with the deuterated CHCs, and also just to compare the cuticular hydrocarbon profile of the spiders and the ants. So the first thing and the first challenge was to go there and actually collect living spiders, which is not a trivial challenge. So the way we did it is we placed dry pitfall traps within a 1/2 meter to 1 meter of 10 different ant nests in the Dalquest Desert Research Station site. And these pitfall traps consisted of 3 cups, one with the set in the soil with the bottom cut out, another cup placed inside without its bottom cut out, and then another plastic cup with its bottom cut out placed inside of that. This cup was to prevent the spiders from crawling up and out of the cup if they did fall in. And then over this whole pitfall, we placed a rock to shadow the spiders and keep them from baking or overheating in the dry desert sun, because presumably the spiders would come out at night and we would have to hike down during the daytime and see if we got them. We were fortunate enough to collect 2 living spiders. That was it, but 2 living spiders, 2 adult females. So this is Norm Horner on the left and myself on the right, holding up our prizes. So we took these spiders back to the research station and we introduced them to a little glass Pyrex dish that had sand at the bottom, and we placed a small piece of wood inside the dish on top of the sand to give the spiders a hiding place. And what we found, first of all, is the next morning after we introduced the spiders, we discovered that the spiders had built these little sandy arenas, these round arenas that they used as retreats. This is a spider inside one of these arenas. When we introduced the spiders to the arena, that evening, we introduced the deuterium-coated ants that we collected from the very same nest that we collected the spiders. We introduced the ants to the dish and we watched the behavioral interaction between the spiders and the ants. And what we discovered is we didn't see any of this pseudo-antination, but rather what we saw is marmicophagy. So after introducing the ants to the container, the spiders within just a little bit over 1 minute emerged from under the bark. So they were able to detect the movement of the ants. They emerged from under their hiding place. They approached the ants. They bit the ants in the rear legs. Then they retreated. And when the ant was paralyzed, which only took a few seconds for the venom to take effect, the spider reapproached the ant, bit it behind the head, and carried it away to feed. And then if a living ant approached the spider that had hold of the dead ant, the spider twisted its body around and presented the dead ant to the living ant like a shield. This kind of marmicophagy, this behavioral interaction between the hunting behavior of the spider and the ant, is almost identical to the hunting behavior published by Stano Pekar for Zodorion. So it's extremely similar to the ethology and to the behavioral patterns that we see in the marmicophagous behavior of Zodorion spiders. Now, for the chemical interactions, this graph compares the deuterium-marked CHC compounds of the ant, the host ants, with the spiders. So the deuterium is on the y-axis, and what you should notice is that the spiders are not picking up the CHC deuterium-marked profiles of the ants. So they're not picking up this deuterated CHC profile. But when Adrian compared the CHC profiles in general of the spider, which is on the bottom, with a CHC profile, the cuticular hydrocarbon profile of 2 different species of Novomessor, Coccarelli and albicetosis, hopefully you can see that there's a great deal of correspondence between the CHC profiles of the spiders and the host ants. So the chemical analysis by Adrian Bruckner found 61 compounds from the cuticle extractions of the ants and the spiders. 60 compounds were detected for the spiders, 42 for one species of Novomessor, 37 for another species of Novomessor. 23 compounds were shared by all 3 species, by the spiders and the 2 ants. And the spiders shared 13 compounds with at least one of the 2 species. 8 compounds were unique for the spiders. So in conclusion, what we found is that Myrmecicultur chihuahuensis is a marmicophage. Its hunting behavior is very similar to that of other species of spider marmicophages, particularly Zodorion. It lives in close proximity to the host ant nest. Maybe it's living inside. We don't know yet. It's unusual that it's only been found in close adjacency to ant nests, but we think it's probably living in close proximity and probably not inside the nest. It does share hydrocarbons with the host ants, which may facilitate hunting. Whether it's producing those CHC compounds de novo, or whether it is somehow picking them up from the ants, that we still do not know. But it shows no other behavioral integration strategy. So once it bites, it retreats, and it's using that dead ant as a shield against any living ants. I'll conclude with some videos. Let me turn the audio off. And these were taken under red light at night, which is why it looks kind of unusual. Just to orient you, the spider is right here in this video, and the spider is right here in this video. They're not great videos, but they'll give you an idea of the hunting behavior. So watch the spider. It rushes at the ant from the rear. It bites the rear leg, retreats, may bite again. It'll bite numerous times, and every ant that we placed in with the spiders was killed. Oops, and in this video, you're going to see the same thing. Let me turn the sound off. So here's the spider. It rushes at the end, it bites the rear. And then you'll see it retreat and wait for the paralysis to happen. So that's it. Short, sweet talk. Thank you.
Thank you so very much, Paula. That was fantastic. Again, if you have any questions, please make sure that you put them down in the chat.
So it takes multiple bites to put the ant down, basically.
I'm not sure, Bruce. If we saw them bite multiple times or just bite once. The venom really doesn't take long to take effect. And if we put 7 ants in the enclosure with the spider, the spiders killed every living ant that was with them. So they seem to use what's called an overkill strategy, which has also been reported in other marmicophages. And it's thought that overkill, where they're killing way more ants than they can possibly eat, is a strategy that is hypothesized to be seen when a spider is hunting a particularly dangerous prey. So if they have the prey in front of them, they'll just like massacre whatever is close by.
Yeah, very different from mimetids.
Very different than mimetids, but very similar to Zodorion.
We have a couple of questions in the chat. Who are these spiders most closely related to?
That's an open question still. I mean, they are on their own branch based on, they're not, in the original molecular phylogenetic study that Martine did, they came out actually not very close to Zodorion. I've been in correspondence with Martine, and he's hoping that we'll be able to collect some more of these spiders and send them to him for a UCE analysis. What he did was a multi-locus analysis, and I think he wants to repeat that and see if we can figure out a closer relative to Marmiciculturity. But right now, I'm not convinced that that any of us quite know where it should be placed.
Okay. So Madeline wants to know, what's the purpose of the first cup in the pitfall trap? Was that just to mark the location?
The first cup with the bottom cut out was in case rainwater got in. I don't think it's necessary because these cups we only put in place very briefly, so only overnight. But what we did, we were at the site for 1 week, and after we left, Norm Horner, who lives closer to Dalquest than I do, he left the bottom cup, the bottom intact cup, in place with preservative. So that bottom cup, it was just there for convenience so that he could use it as a longer term wet pitfall trap.
One more question. Do they hunt ants exclusively? Did you see any other dead insects or debris?
So this was such a shock to us because we really expected these spiders to be eating other arthropods and not the ants. So we were not expecting them to hunt the ants. I do not think that they eat other prey. I think they're exclusively myrmecophages, much like Zodorion. But I'm not 100% certain of that, but the fact that we only see them and collect them in vicinity of ant nests and this very specialized hunting behavior that we see that is so similar to other obligate myrmecophages, all of that suggests that ants are the only prey.
Is there anything else you can tell us about the sand retreats?
About the what?
The sand retreats?
Oh, no, that was another surprise, a pleasant surprise to see those little arenas. They were not complete sand retreats. So if you know of Zodorion makes a little igloo retreat that's completely enclosed. But these retreats, I do not believe, these spiders were in the container long enough that were they to build a complete igloo-shaped retreat, they would have done so. But they didn't. They just built those little sandy arenas, those open arenas. Okay.
There are just two more questions. So the first one is, do you know if the spider attacks soldiers and workers similarly? Because it seems like in the videos, they were preying on larger ants.
Yeah, for Novomessor, that's an interesting question, because what I would like to do is to really see if there's a difference in hunting strategy between when they hunt Pagonomyrmex and when they hunt Novomessor. Because Novomessor tends to have, it tends to be a much more calm species. So it's not a particularly aggressive species. We have done little pseudo introductions of Novomessor from one colony and plop them onto a different colony. Nobody cares. No one is attacked. So Novomessor seems to be a very low-key, relaxed kind of ant that does not seem to have the morphological divergence in worker versus in any kind of worker castes. Whereas Pagonomyrmex does tend to have a generally different caste system and caste sizes, and Pagonomyrmex tends to be a much more aggressive species or aggressive genus. So it'd be interesting to see if there's a difference in hunting strategies between of the spider between those two different genera. But there's not very much of a caste or size difference.
Okay. Bruce had a follow-up question. What will the immature spiders feed on?
I don't know.
Like I said, I kind of tell you what the chat was about. So a lot of recommendations of if you could rear them, that's probably a way of answering a lot of these questions, especially the hydrocarbons.
Oh yeah. Although, I mean, I don't pick easy groups to study. So the fact that we got 2 living spiders was fabulous. I mean, an N of 2 was like, we were so excited to have 2 spiders in our pitfall cup. So the idea of getting enough spiders to rear in the lab, sure, that would be great. Whether we can do it, I don't know. We're going to try. We're going to continue this study, and Norm and I will travel back to Dalquest to repeat this dry pitfall trapping.
Thank you so very much, Paula.